Hi, welcome to the screencast. This is for blood. I don't really ask a lot of questions about the functions of blood within the larger context of the body, but it's one of the more central, what's the word I'm looking for? One of the more central tissues or systems within the body. So sort of like your nervous system controls and monitors a lot of things, your bloodstream connects physically lots of different parts of your body together, right? So it delivers oxygen from the lungs to all of your tissues. It picks up nutrients, it picks up nutrients from the digestive system and distributes that, and then it brings metabolic waste like CO2 to the lungs and nitrogenous waste and other metabolic byproducts to the kidneys or the liver for elimination. In addition to that, it distributes all of your hormones. So remember, we just finished the endocrine system. All of those hormones end up in the bloodstream, and that gets all of these very important signals everywhere. So I don't really focus on these things on the exam, but it really is one of your central systems. The blood itself doesn't regulate body temperature so much, but your vascular system does by controlling where the blood goes, whether it's close to the surface or stays more internally. This helps you distribute heat. Your bloodstream is one of your primary liquid components, and it helps keep your whole body pH neutral. And then it's got cells and proteins within it that help protect against blood loss, and then it also carries the cells that make up your immune system. So your white blood cells are primarily what your immune system is. Sorry, I keep on having to pause it to clear my throat. I don't know what's wrong with me today. Let's see. The important thing on this slide here is just this part. Remember that number. I don't generally have you all commit a lot of numbers to memory, but 7.35 to 7.45 as the normal range for optimal pH you want to know. And if your bloodstream gets much beyond that, your body becomes rather unhappy. So the pH is for the plasma, that is the liquid portion of your bloodstream. Then you have what we call the formed elements, which is your red blood cells, your white blood cells, and what we call platelets, which are just cytoplasmic fragments, and that's why we refer to these three things as the formed elements as opposed to your blood cells, because the platelets are not proper cells. Hematocrit we will go over in lab, but we also want to go over in lecture. So if you're going to do somebody's hematocrit, or calculate it, measure it, you take some of their blood, you put it in a tube, kind of like this one, and then you put that tube in a centrifuge, which spins the blood around really fast, and it's like applying, well, it's applying G-forces to the bloodstream, magnifying the effects of gravity so that the heavier things settle to the bottom. Your red blood cells are packed with hemoglobin, so they're dense. And as you were going to learn, that hemoglobin contains iron, which is heavy. So your red blood cells settle to the bottom, the white blood cells and the platelets, which is called the buffy coat, on top of that. I have no idea where that term buffy coat comes from, but I just like it. And then the plasma settles on top because it's not as dense as any of your formed elements. So then what you do for the hematocrit is you say, okay, if this is 100% of your blood volume, how much of your blood volume is just red blood cells? So you're just measuring the percentage of the volume. So this is another instance where I will have you commit 42 and 47 plus or minus 5 to memory for females and males respectively. So first we are going to look briefly at what is in the plasma. We don't really have time, the way this course is structured, to spend a lot of time on blood. There's a lot more going on in it than we have time to cover. So the plasma portion is mostly water, as it is a liquid. It does have a fair amount of protein in it. It's a lot of a protein called albumin, and that's in part so that the plasma has the same osmolarity as the cytoplasm of all of the cells floating around with it, right? If the plasma were too watery, then all of your blood cells would swell with water because of osmosis. And that's probably the most important part, is to know that it's mostly water, but that it's got a lot of protein in it so that it is isotonic to your cells. The plasma is going to contain electrolytes. You may recall that they are things like sodium, potassium, and anyone? There's no one there, so I'll just say it. Calcium, which is bivalent. It's got a charge of positive too. And then your respiratory gases, oxygen, and CO2, plus hormones. Then you have your formed elements. Again, red blood cells, white blood cells, and platelets down the bottom. We will cover them in this order. First up are your red blood cells, otherwise known as erythrocytes. Everything about them is specialized, starting with their shape. Biconcave just means if you looked at a red blood cell from the side, and I'm exaggerating a bit, right, it would look like that. That almost looks more like a mushroom. But you can see it's concave here and concave there. What this does is give it a very big surface area to volume ratio, so that if you are a molecule of hemoglobin anywhere in the cell, you're never very far away from the surface of the cell. So imagine you took the same cell and just blew it up big like a circle. Now, if you have hemoglobin in towards the middle of the cell, these molecules here of hemoglobin are very far from the surface. And your red blood cells rely on diffusion for the movement of oxygen in and out of the red blood cells. So you want all of your oxygen-carrying hemoglobin molecules as close to the surface as possible. A nucleate here, you might guess, means without a nucleus. So they are essentially bags of hemoglobin. They have very few organelles and no nucleus, simply so that they can transport respiratory gases. So hemoglobin, we want to know a little bit about now, and we'll talk more about hemoglobin when we cover the respiratory system, and we'll talk about oxygen and CO2 and how they interact with hemoglobin. So for now, know that this is here one molecule of hemoglobin, but it contains 1, 2, 3, 4 peptide chains. So if you remember from Anatomy and Physiology 1, that's called quaternary structure, 4 peptides. Each one of these peptides has a heme in the middle, or it's labeled here. That heme is this ring structure here called porphyrin, which you don't need to know, with an iron in the middle, and it is the iron that carries the oxygen. So if you leave something metal, steel, outside, and it's not stainless steel, it will oxidize. The iron in that steel will react with oxygen in the air to form iron oxide or rust. So because iron likes to interact with oxygen, it is used as the oxygen carrying element or component in your hemoglobin. So then, because you have 4 hemes in each hemoglobin, and each heme can carry a molecule of oxygen, each molecule of hemoglobin can carry up to 4 oxygens. And that, again, is going to become a little bit more important when we get to the respiratory system. This slide we're just going to skip, because it's just, what's the word I'm looking for? More vocabulary, and you all don't really need more vocabulary. So we next want to talk about the production of red blood cells, because this is kind of where we are. but we have to take one step back and talk about blood cell production in general, which is called hematopoiesis, that's blood cell formation. And you want to know that it occurs in the red marrow. So this is the marrow that's in the spongy bone of your skeleton. So you also want to know that it's this one cell type here called a hemocytoblast that gives rise to all of your formed elements. So you've got your lymphocytes, or your white blood cells, lymphocytes, monocytes, all these cells here, I won't name them all, red blood cells and platelets, which are actually fragments of this cell type here called a megakaryocyte. But developmentally, they're all derived from the same stem cell. So what would happen is this cell divides. One of its progeny is going to become one of these cell types, and then the other one will divide again, and one of its progeny will become one of these cell types, and then the other one will divide again. So this is what stem cells do. They divide, and then one of the cells from that division takes on a different developmental fate, and the other cell remains behind to keep dividing again. So it's a self-replenishing population of cells. And this is one of the examples of adult stem cells that people are experimenting with so they don't have to use fetal stem cells. Next up then is back to red blood cells and the production thereof. So red blood cell production is called erythropoiesis. So the poiesis, it is a Greek term. Some of our terms are Greek, some of them are Latin, some of them are French, and some of them I don't know where they come from. So erythropoiesis is under the control of erythropoietin, which is produced by the kidney and is triggered by hypoxia. You could probably guess hypo means less, oxia refers to oxygen. So this is low oxygen levels in the bloodstream. When that gets detected by the kidney produces EPO. When the EPO or erythropoietin makes it to the bone marrow, it stimulates the production of red blood cells. And EPO technically is considered a hormone, even though it's not produced by a traditional endocrine gland. And this is why I don't really have you all, one of the reasons why I don't have you all memorize the definition of a hormone, because there are so many things that don't come from endocrine glands, but that act like hormones, or are hormones. So hypoxia is next. As we said, this is low oxygen levels. It could be low oxygen levels in the blood so that your whole body is hypoxic, or it could just be a lack of oxygen to an individual tissue. So if you sleep on your arm wrong and you cut off blood flow to your arm and it falls asleep, that would be hypoxia. I think that's why it falls asleep. It might also be neurological. I'm not an MD, so don't quote me on all these things. But lack of blood to any individual tissue would cause that individual tissue to become hypoxic. You could just have less oxygen in your bloodstream, maybe because you're at altitude, or any number of reasons. There could be something wrong with your blood, like you're anemic and your oxygen carrying capacity is lowered. So you just want to know that hypoxia is a lack of oxygen. Many things can cause it, and individual tissues can be hypoxic, or your entire body can be hypoxic. So then, your red blood cells do not live forever. I think they are the shortest lived of all of your blood cells. I don't know about them versus the platelets. I'm not sure how long platelets survive. But your red blood cells do not last very long. You have a lot of them, so they have to be constantly cleansed from the bloodstream. Most of them are phagocytized in the spleen. That is the primary function of the spleen. It does some other things, but it removes red blood cells. So you want to know regarding red blood cell destruction, where they get destroyed or removed from circulation, which is the spleen, and then what happens to the iron, what happens to the heme, and what happens to the peptide globin chain. So as this here says, the iron is salvaged for reuse. It is going to be stored in the liver and then eventually sent to the bone marrow and incorporated into new hemoglobin. So it just gets reused in its original purpose, or for its original purpose. That porphyrin ring, the rest of the heme itself, is degraded and ends up in the bile as a pigment called bilirubin. Bile is secreted by the liver into your small intestine, so the bilirubin comes out with your feces and is primarily where your feces gets its color. Without the bilirubin, feces would be much lighter because it's mostly bacteria, which is not all that dark. Generally speaking, bacteria are not all that dark. Then the globin, you will remember, is a peptide chain. So it just gets digested down into individual amino acids, and then they get incorporated into new proteins or recycled into different proteins. Then we have just a couple of erythrocyte disorders, or just one, anemia. So anemia, as this says right here, is low oxygen carrying capacity in the bloodstream. It can be caused by lots of different things. So there are different kinds of anemia, which we will cover. So anemia could be caused by insufficient erythrocytes, so not enough red blood cells, maybe because you're losing blood, or maybe your red blood cells are rupturing, as happens in malaria, or you're not producing enough red blood cells, which would be aplastic anemia. Then you could also have low hemoglobin content. Iron deficiency, as far as I know, is the primary reason why people would have low hemoglobin content. And then you can also just have problems with your hemoglobin, which is referred to as thalassemia. And for reasons that I don't totally understand, the problems in the hemoglobin itself, this is going to be in the peptide part of the hemoglobin, also affect the shape and hardiness of the red blood cell. So in the case of sickle cell anemia, which would be a thalassemia, there is a mutation in the gene that causes one amino acid substitution. It's a lysine, I think, instead of a threonine. I used to know, but one amino acid in the whole protein gets changed. That changes the shape of the hemoglobin and also changes the shape of the red blood cells, so they go from this nice biconcave disc shape to this sickle cell. So the misshapen hemoglobin is not as good at binding oxygen, but also the cell itself is misshapen, which gives it the wrong surface area to volume ratio, and also makes it brittle, so the sickle cells are more likely to break apart when they're passing through small capillaries and they undergo some shearing forces. Then we get to the white blood cells. This I kind of consider the meat of the chapter, but it's all important. So the first thing you want to know is the difference between your granulocytes and your agranulocytes. So just be able to match the cell type with agranulocyte or granulocyte. Granulocyte means that the cells have clumps of protein in the cytoplasm that we call granules, and then the monocytes and the lymphocytes do not have granules, so we call them agranulocytes. And you're going to see that the three granulocytes are named for the kind of stain that their granules absorb. Let's see. Oh, here's a couple things I forgot to mention. This term right here, if you had me for Anatomy and Physiology 1, you know what diapedesis is. This is when white blood cells attach to the cell wall, crawl out of the bloodstream and migrate towards a wound. So this is not cells are under pressure in the bloodstream and they get squirted out like water coming out of a garden hose. This is, I don't want to say intentional because your cells don't have intentions, but it is directed movement where the white blood cell attaches to the blood vessel wall, makes its way through the basement membrane, maybe causes a couple of epithelial cells to move apart so that it can get through, and then moves towards a wound, primarily due to chemoattractants that are released from wounds. So then, if you do have an infection, you're going to want lots of white blood cells to help fight that infection. So your white blood cells undergo mitosis, and we call that leukocytosis, which is just a high white blood cell count. Don't worry about the number, just know that it's high. Let's see. So first up, the 3 granulocytes. Neutrophils. For lecture, you just want to know what's in the granules, which is enzymes called defensins. Well, actually, it's 2 things. It's hydrolytic enzymes and defensins. The defensins, what they do is poke holes in bacterial cells, and then the hydrolytic enzymes start digesting the bacteria, and then the neutrophil itself will phagocytize the bacteria that it just killed. For lab, you want to remember that they're called polymorphonuclear leukocytes. This means many-shaped nucleus. And you can see that this one here, right, this is the nucleus, and it's got this really weird shape to it. Each neutrophil looks a little bit different. All their nuclei look kind of like the Rorschach ink blot tests. So that's the neutrophils. Then the eosinophils, you want to know, attack and digest parasitic roundworms. So they just dump hydrolytic or digestive enzymes on the outside of the worm, and then the little digested bits of the worms are phagocytized. These are called eosinophils because the stain that their granules absorb is called eosin, which is named after the Greek god of the sun, Eos. And the neutrophils, I forget if the neutrophils are called neutrophils because the granules stain neutral, which means the same color as everything else in the cytoplasm, or if the pH of the stain that they absorb is neutral. I should look that up, but you don't need to know it, so it's not super important. Then the basophils, their granules contain histamine. They're called basophils because the granules, or the stain that the granules absorb, is called basic fuchsin. And it is the purple stain that gives the granules their purple color. And histamine, you will learn later, is an inflammatory chemical. It's also involved in allergies, but allergies are kind of a loss of homeostasis. They're a bit of an overreaction to things, but histamines are also involved in your normal, healthy, inflammatory response when you do have an infection or tissue damage. And inflammation actually helps fight infection and tissue damage because it brings lots of blood to the site of injury. Then we get to our lymphocytes, which there are 2 different kinds of, and we're not really going to worry about the 2 different kinds for right now. Just know that some of your lymphocytes are really good at killing off cells that are infected with viruses, and some of your lymphocytes produce antibodies, which are proteins that your immune system makes that just labels foreign things as foreign. Then you have monocytes. So these cells are especially good at diapedesis because it is their job to crawl out of the bloodstream into other tissues, where we then call them macrophages. So when we cover different tissues, you are going to see that it's macrophages in the spleen that are clearing dead red blood cells. and macrophages in the liver that are helping to cleanse the blood, and macrophages in your lungs that help keep your lungs clear of foreign debris. So any dust and pollen and stuff that you inhale, some of it is digested by your monocytes. Some of it ends up just being cleared from your lungs in the mucus. So that's the monocytes. And this is really how we're going to think of them here. We can't know everything about each cell type. So we're going to say monocytes are macrophages, and we'll talk later about how macrophages help activate lymphocytes. For now, we're not going to worry about it. Leukopoiesis. This is the production of white blood cells, and there are two groups of chemicals or families of sort of developmental signaling molecules, interleukins and colony stimulating factors. So when those hemocytoblasts are exposed to different combinations of interleukins and colony stimulating factors, that is what directs the development of one white blood cell type versus another white blood cell type. So just remember those two terms. Don't worry about any of the numbers here. There are more interleukins than I can even remember. But also do recall that all of your formed elements come from the hemocytoblast. You know what? We're not even going to worry about this. Just cross it right off. I never ask questions on the exam about it. Yeah. Then platelets. So you will recall that platelets are not actual cells. They are little blobs of cytoplasm that still are contained within a membrane. It's just not a complete cell. So they're little membrane-bound sacs of cytoplasm from the megakaryocyte. It is the hormone thrombopoietin that stimulates the production of platelets. And their job is to form what is called a platelet plug. Sorry, that's supposed to be an underline, not cross out. And this is what happens before clotting or coagulation as a temporary blood loss measure before the blood vessel can actually start healing. This then brings us to hemostasis, which is 3 steps outlined here. So for each one of these, or for hemostasis, I should say, you want to know the names of the steps, the order in which they take place, and a basic description of each one. Vascular spasm is easy enough to understand. As you can see right here, the open end of this blood vessel is starting to pinch off a little bit. That reduces the amount of blood loss that's going to take place until this blood vessel can repair itself. Then the platelets are going to stick to the connective tissue in this part of the blood vessel, which is normally not exposed, and then when they stick to that, they release chemicals that attract other platelets. Those platelets release more chemicals that attract more platelets. That's from AP1, you may remember. One of the examples of a positive feedback loop. And then coagulation is the formation of a clot, which is composed of fibrin. So please do keep straight the difference between hemostasis and coagulation. Coagulation is the last step in hemostasis. So if I'm asking about hemostasis, it is just 3 things, and it does not necessarily include all of the details of coagulation. Don't worry about this. I don't know why it's even still there. I thought I deleted it. Then you have coagulation. This is the formation of a fibrin clot. This is something that you need to know the details of, although not all of these nitty-gritty details over here, but the details over here. So there's 2 mechanisms of activation, the intrinsic system and the extrinsic system. This here says damage to a blood vessel wall, but this is where the platelets attach to the blood vessel wall, and then the platelets release attractive chemicals that attract more platelets. So it's triggered by exposure of the blood vessel wall. That's the physical trigger, but chemically it is PF3 from the platelets that's setting off this sort of biochemical cascade, if you will. And then the extrinsic pathway is triggered when cells that are not part of your blood vessels are ruptured. So there's a whole lot of stuff inside of your cells, and it's supposed to stay inside of your cells. So whenever a cell spills its guts, there's all sorts of stuff that leaks out into the bloodstream or the extracellular fluid that your immune system and other parts of your body can detect to let the body know when it's been damaged. So one of these things we just call tissue factor, and that can also activate this clotting cascade. So you'll notice that both of these pathways intersect right here, and that's where we're going to pick up. So you just need to know you start here, and then you skip all that crap and go right down to there, which is what the old Merib textbook calls prothrombin activator, what the OpenStack's book calls prothrombinase. So prothrombinase is an enzyme, or as this says here, an enzyme complex, so like a group of enzymes. Enzymes catalyze reactions. They make things happen. So what prothrombinase does is catalyze the conversion of prothrombin to thrombin. Prothrombin is a proenzyme, which means it's inactive, and thrombin is an enzyme in its active form. What thrombin does is catalyze the conversion of fibrinogen to fibrin, so this part here. Fibrinogen is soluble, which means it's happy floating around in water. Fibrin is insoluble, so anything that's insoluble floating around in the water will automatically stick to something else that is insoluble. So all the fibrin molecules stick to each other and form a big mesh that other things then get caught in, like red blood cells. So it's sort of like a spider web. So again, this is prothrombinase. It's an enzyme that catalyzes this reaction of prothrombin becoming thrombin. Thrombin is also an enzyme which will catalyze the conversion of fibrinogen to fibrin. I don't know exactly how this is achieved, but I would imagine fibrinogen is going to be folded up properly as a protein with all of the hydrophilic amino acids on the outside, all of the hydrophobic amino acids on the inside, and perhaps thrombin clips off a little piece or adds something to the fibrinogen, which causes it to change its shape, and then that exposes its sticky hydrophobic interior and basically turns the protein inside out. So it's almost like purposefully denaturing the fibrinogen to make it into this sticky fibrin stuff. And that's the end. That's a whole chapter in 33 minutes. Cool. Thanks for sticking around this long.